This unit is called the unknown assignment, is going to be covering putting together the diagnostic media and some how you read test results with a report. So what we do, I'm going to click on where it says documents of the unknown assignment. If you're in a research laboratory, there are times where you want to figure out if you find an organism, you want to run some tests on it. That's what the diagnostic tests are. What we're going to do with this assignment is you're going to be given a set of information and you're going to have to figure out what is your bacteria type based on these test results. Now, in a lab setting, you would be given a test tube, an auger slant that contains a bacteria type, and your test tube would only have a number on it. That's it, just a number. You would write your number down. You would then, the first thing you would do is you would do a gram stain to see what is the gram reaction and then what are the cell shape arrangements. And then from that information right there, you would take our flow chart and figure out what series of tests are you going to run. I'm going to go through all these documents with you. Let me go ahead and open. In fact, I think I already had this. This is the unknown flow chart for microbiology lab. That is this one. It is getting a little old, and you still can read it. I tried to write in here the red broth. I'll go through all of this as we go through the test examples. But let me go back. So I'm going to open up this document first. So instructions for completing the unknown. Now, we are not in a lab setting. So basically, I have emailed you what I call the unknown information sheet. And it's for you specifically. You get your own bacteria and it has all the information on that sheet you're going to need to complete this exercise. So starting here first, be sure and record your unknown number. I'll show you that in a second. Assume your unknown is a pure culture. Okay, well, I'm giving you a sheet for this. You will need to perform a gram stain. So you would be given a test tube that's an auger slant, and it's going to have some bacteria growing in it. First thing you would do is a gram stain. The gram stain reaction and cell shape arrangement are provided on your unknown information sheet. So refer to that, and I'll show you. I'm going to go through two examples of that with you. Note your unknown's gram reaction, cell shape, and arrangement. This is all part of the format you're going to be doing. According to your gram stain results, determine which portion of the flow chart you will need to follow. Now, on the flow chart, you can see one side is for gram positive. The only way you would know that is if you do a gram stain. If you scroll down, the other side is for gram negative. So you're only going to be using one side of this flow chart. Depends on what your gram reaction was. Now, the test results are provided on your unknown information sheet, the one that you had emailed to you. Refer to that for all the tests that were run. You will need to include the following information in your unknown report. So see the sample unknown assignment posted under this document for the correct format. And this has some of the format as well. It's talking about this, a sample unknown. I'll go through that with you in a second also. You will need to include the following information in your report. So a heading that includes your name, the instructor's name, the date, and microbiology lab, the gram stain results, cell shape arrangement. Now look, that one, just do your best. And look, the cell shape, you should be able to tell pretty easily. The arrangement, you go with what's the majority, but you can't really get that wrong. The gram stain, you need to get correct, and I think with the test results, you'll figure that out. But a list of the tests performed, I'll show you that. And then basically you have to explain each test and go into detail. Like, what is this test testing for? Is it testing for an enzyme? Do you have to add reagents? That kind of thing. And then you're going to answer some research questions, and you can use the internet or any source for that. And basically, what family does your bacteria belong to once you figure out what you have? Translation of genus name and origin. Some of these actually translate. Klebsiella, a bacteria type, was named after the scientist. The genus discovered it. The species name, which is the second part of that. The genus is capitalized. The species is lowercase. Major habitat where it's found, Where do you find it? If you don't find it in the habitat, find where it might be located. And if it causes any disease, not everything does. Some things cause food spoilage. When you finish your report, click on the link that says Submit Your Unknown Assignment here under the Unknown Assignment tab. You turn in your report there. It will be a Word document. I'm going to go back through the breadcrumbs briefly. See where it says here. Click here to submit your unknown assignment. Due date, points possible, and basically you will have your report saved to your computer, and this gives you options, and obviously you're going to be probably browsing. The text submission, I'd prefer you use a Word document, and then you're going to hit submit. So it's pretty easy. That's where you will turn in your assignment. So going back to this, all right, so we're on this instructions sheet. Now, you do have to cite on these research questions at the very bottom, cite where you get your information from. Examples of how to cite sources. If you use a book, you can do this. The internet, most people do. Wikipedia, I'm not sure I would use that. Try to use a reliable source, but you can actually just do it this way if you wish. Just have the link provided in the website. Follow this format when submitting your unknown report, and we're talking about the format below. Part of the grade for this report will be following this format. And I'll go back to your submission. There's a rubric for this assignment if you want to look at it. Follow it exactly as below. When describing anything, use as much detail as necessary without it becoming a dissertation. So when writing the name of the bacteria, use the correct procedure, and I'll show you that. So here's how you're going to do it. So you have unknown assignment title, your name, my name, microbiology lab, date. Put your unknown number. Make sure you bold it and underline it. That way I can see it. Your gram stain results, just like this. Have a little title here and then put your gram stain underneath. Cell shape arrangement, same way. Do it just like this and then have the description underneath. Like I said, this is, you can't get this wrong. Put what you think it is. But from the pictures I gave you, sometimes it's hard to tell. And it's hard in a microscope to tell. Make sure to name the cell shape first, then write the arrangement. So cells were rods arranged in chains, indicating the bacteria cell shape was a bacillus and the arrangement was a streptobacillus. So a chain of rods. List of tests performed. List them in the correct order as it is on the flow chart. Like this, for instance. Description of test results. Now here's where you want to go through and describe in as much detail as possible. What is the EMB plate? What are you testing for? For instance, EMB was positive because it showed metallic green bacterial colonies on the agar. This means the organism is a lactose fermenter and a coliform. The SIM plug shows no motility but was positive for endo production. The test for endo production, Kovacs reagent must be added. And of course, with all these tests, you have to assume they're all final. There's no reagents for us to add. So all the things I'm giving you, everything has been added in advance. Just kind of assume that. And it says that somewhere in here, but just assume that all of these are final results. Urease, so just basically your test, describe what the test you're testing for. Is it an enzyme, any reagents involved, and what does a positive mean or a negative mean? So the identification of your unknown. Now, you can write this two ways. You do it like this. This is Mycobacterium tuberculosis. You do two separate underlines. You capitalize the genus name, and you lowercase the species. Or you can just do it all in italics, but you do have to capitalize the genus and lowercase the species. That's how you write. That's correct scientific notation. So remember to underline separate genus and species, and I'll put in there also to capitalize the genus name. Research questions, 30% of the grade. What family does your organism belong to? Mycobacteriaceae, which that one, it says, word is saying, oh my God, what'd you put there? It's not spelled, but family names do not have to be underlined or italics. Translation of genus name or origin. The genus name is Latin. It refers to Thor playing Fortnite. I don't think that's actually accurate, but it will say this. Go on the internet. We usually have a thing that we use. It's a microbiology standard, but it's a book. It's in the library, and nobody has access to that right now. So look up the genus name. See if it has any meaning whatsoever. Species name, and I didn't put here much, but yes, of course, if there's a name and origin, include as much detail as possible. What major habitat does your organism belong to? Is it found in skin, dirt? Does it grow on burned tissue, anything like that? And then any diseases caused. And the blue gives you instructions for all of that. But yeah, put anything there are certain things that cause disease. I mean, Mycobacterium tuberculosis causes tuberculosis, a lung condition. Not everything causes a disease, but put anything if it causes food spoilage or any problems at all. And when you see this, I'll put the example on the bottom here. I'd like you to put where you got your information below this. And when you see this video, or when you see this document, okay, it'll be down there. So these are the instructions. And then what I did, I'm going to go ahead and close this out. The sample unknown, same thing. I will put the how you cite your web page. Oop. Didn't want to do that. So this would be like the real thing. So here is, I know before I had it in the middle. This is fine here. But here's the name, the number, gram stain, cell shape arrangement, list of tests performed, description of test in there, identification of unknown, of course this one's fake, family name, translation, genus name and origin, translation, species name and origin, major habitat, disease caused. So let me show you how you're going to get here. If you're wondering, how are we going to figure out what our bacteria is? Okay, we'll do that next. I'm going to keep this just one video. So here is a sample sheet that you have sent to you. It's in your email. So if you haven't checked it yet and you're watching this video, go check. You have a unknown assignment sheet there for you. Here's the number. Your unknown number is 66, just like the one that wiped out the Jedi. Gram stain reaction, cell shape arrangement. Now this is the picture you're going to use for that. First of all, what color is this? Well, it's definitely, and I'm talking about the cells, not the background. Definitely blue, purple, so it's gram positive. Now you can, these Word documents, if you wanted to get super, super close, you can tell that's a rod, okay? That's not circle shaped, even though like, oh my God, there's a circle. No, the majority, in fact, here's a couple that are joined together. But look, the majority of them are all rods. And to be honest with this one, most of this, and you go with the majority, as far as the arrangement goes, these are all by themselves. They're singles. That's the word you would use. They're isolated. There are two that are connected. There's two here. And in fact, this even looks like, now they kind of blew that up a little bit to make it look larger, but I would say the bacterial cell shape for this is a bacillus. It is gram positive because it's purple or blue. And the arrangement would be singles or isolated. If they were all like that, the majority were in pairs, I'd say a diplobacillus or something like that. You can refer back to the cell shape arrangement section. It was in Unit 1, and we talked about like what is a cell shape arrangement, that kind of thing. So what you would do is once you figure out your gram stain, so this is a gram positive, so you would then walk up to the front of the class, your gram positive rods. You see that? See, if you're a gram positive Kasai, circle shape, you're going to be on this side. So here are all the tests that you would need to run your unknown experiment. You're going to do a catalase test, that's the bubbling. And you can refer back to the diagnostic media unit for all of this if you need to. I tried to put this in here. It used to just say mannitol. This is the red broth, the mannitol with the little tubes inside. So you need one of those, and then this is actually pretty easy. You would need one citrate, basically. And, well, the first thing you would do is a catalase test. You can actually do that immediately and see if it's plus or minus. Now, catalase reaction. What does that look like to you? That's some serious bubbling. That's a positive. So, you know already, so since you're positive, you're going to be on this end of the flow chart. Oops. So what you would do next is you would get a red, phenol red mannitol fermentation broth with a Durham tube and a citrate slant. So you'd have two things. You would take them back to your table. You would then use your original culture unknown that was given to you to do the gram stain, and then you would inoculate these two sets of media. You would inoculate a red phenol red broth tube and a citrate slant. Do them all at the same time. You would then take those tubes, you would put them in the incubator, wait 24 to 48 hours. You would then come into the lab and read your results. So what are the results? Okay, we already did the catalase. So your results are the phenol red mannitol turned bright yellow. Now it starts out red. Okay, that's why it's called phenol red. Citrates, which are green when they're sterile. And remember, with citrate, blue is positive. So that's definitely a negative. So you have a positive here, and you got a negative here. So you go back. So we already did the catalase. You don't have to let that incubate. Now, your phenol red broth was definitely positive. See, it says strong plus. Once again, you can refer back to the diagnostic media unit. If you don't remember, with the phenol red broths, and I put this at the bottom of the page, there is what is called a weak positive, which is what that's referring to, reaction. And the broth is red, but if it's a weak positive, it turns orange. So if you were to look at your broth tube and it was orange, you would have stayed over here. See, you would have followed down and looked at your citrate. There's the plus, there's the minus, and you would have figured out what you had. But yours was definitely a strong positive. See, it's glowing bright yellow. That's not orange. So, strong positive, and then of course, there's really only one way to go. But look, the citrate was negative. You saw that. Oop, wrong page. That's my other example for you. Okay, this is green. If it was positive, it'd be blue. So what is your organism? You have Corynebacterium. All right, that's the identification of the organism. What you would do then next is you would describe all the tests you did in that section that says description of test. And then your research questions would be based on Corynebacterium. Let's do another example. I'm going to get rid of this. Got a lot of, well, I'm going to leave that actually. Let's do another one. So here is your order. Your unknown number is 666. So we got some Satan involved here. Here's the gram stain reaction. It says is below. And like I was saying, in a lab, you'd be given a test tube like that. You don't know what it is. It'd have a number on it. And your goal is to figure out what's the microbe growing in the tube. Like I said, you can blow these up if you need to. You can get really close. Now, look. Hope you can tell those are definitely red to pink cells. And look, they are rod shaped. See how long and skinny they are? And same thing here. Now, there is a type of arrangements called palisading or palisades. That looks a little bit like that actually. And look how they're all stacked. And then so, look, you can see the majority are. So this would be palisading, definitely. Or pelicides, even. If you were to put that these were also in singles, you would have got credit for that as well. Look, go with the majority. The main thing you want to get right, you definitely should be able to get the cell shape right and the gram stain. So you are actually gram negative. Okay, so what does that mean? Well, that means that if that was your microbe after your gram stain, then you're going to be on this side of the flow chart. So what you do here is gram negative rods. There's not really an option for Cossi for this one. So you're going to need a few things to run your unknown test. You're going to need an EMB plate, okay? Remember the auger plates. The indole, if you think offhand, what is that? Those are the little SIM plugs. Once again, the diagnostic media unit, I went through that. You can add Kovacs to test for indole, motility, the black hydrogen sulfide. And you can see here, look, like this says H2S plus or minus, H2S, this is plus, this is indole positive, negative. So you're going to need an EMB plate, a SIM plug, which you use the little needles for those, you stab it down the middle, a urease broth, and a citrate slant. So you're going to need 4 things. So you would grab all 4 of these diagnostic tests. You would then head back to your table and you would inoculate from your culture tube. So basically, I'm going to go back. So you would take, each time, do aseptic technique. You would streak an EMB plate. Oop. Let me go back to you would streak a plate. You would use one of the needles, and I covered this with the SIM. You would just kind of stab these down the middle. Okay, so you'd streak a plate first, put that down. You'd pick up a SIM gel plug. You would use aseptic technique. You'd go back to your culture. You'd get some on the needles, stab it down the middle. You'd put that one away. Pick up the urea broth. Remember, those are salmon colored. You would inoculate the broth like you would any kind of broth tube. You'd put that away. And then you would streak an auger slant. So you do all 4 of those all at once. You would label everything. You'd put the tubes in a rack. You'd put your plate maybe on top of the rack. You'd put everything in the incubator. You let it sit for 24 to 48 hours. And then when you came back 2 days later, you would then look at all your results. Now remember, the SIM, you have to add Kovacs to that to test for indole. So here are your test results. Now, and like it says here, assume all reagents have been added, all results are final, meaning you don't have to add anything to them. So everything you're looking at, all the proper reagents have been added. You see the EMB plate? Now look, those are dark colonies on the EMB. So that is definitely a positive. If it was negative for lactose, it almost looked like the same color that you started with. You wouldn't hardly see anything. If it was green metallic sheen, that's also positive for lactose because that's a coliform. Coliforms are also lactose fermenters. You would then look at your SIM. So this one you would add Kovacs, remember, to this, and you could tell 3 things with the SIM: sulfur, indole, motility. You can see if it produced hydrogen sulfide, if it's modal, or if it produced indole or indole. In this case, you have to assume all the tests are final. So here's the SIM. Do you see a red ring? I do not. So that is indole negative. And with these tests, same thing. You go with the majority. If you have 3 tests that say you have 1 organism and 1 test that says you don't, you go with the 3 that says you do. So the indole is negative. So the lactose is positive. You look at the urease. Now, in this case, this one says they're both negative, and that, as far as the flow chart, that's a definite negative. Remember, urease is kind of salmon colored. It would be pink fuchsia if it was positive. And then you would look at the citrate just for the final conclusion. You can see the blue slant. I know this is green, but that's definitely a positive. Definitely a positive for citrate. Oops, and I meant to click on the other. So, see that means your organism is Klebsiella pneumoniae, and Klebsiella is actually named after the guy that discovered it. So that's how you use a flow chart. So once you get your unknown, you're then going to go back, and I left this open. You're going to fill out all your information just like this. Do the same format. Do it just like that with the headings and the bold and everything. Here's your list of tests that you ran. Here's the description of what the tests were looking for. There's the identification, and then here are the research questions. And like I was saying, please cite where you got your information. When you see this video, there'll be something down here like a citation that I'd like. All right, so that's what you're doing with this unit. You're going to be performing an unknown. Obviously, you've watched this video, I hope, but here are the instructions that I clicked on earlier. Here's the sample unknown. Here's where the flow chart is. Okay, all that is under I call it the documents for the unknown assignment. This video will be above that. And then when you get ready to turn it in, here's your link you click on. And there is a rubric. You can click on it there. A rubric is basically the grading scheme. Look, you're graded on 5 areas: the format, the list of tests performed, the description of your test, did you get the correct identification of the unknown, and then the research questions. How did you answer those? Did you do a good job or a poor job? And yeah, the categories are excellent, fair, poor, and very poor. And that's the grade distribution and breakdown for, it's 100 points total. You can view that anytime. And when you get ready, yeah, you're just simply going to save, like let's pretend. So here's my report. Save as, and I don't know what it's called. Let's see. I have, okay, sample unknown assignment. I'll just save it under my documents. You would have it named, of course. You could put your name it whatever you want the file. And then when you're under the tab that says click here to submit, you would simply browse your computer. Let's go to documents, and there it is, the one we just saved, sample unknown. Hit open. You can see it right there, and then you can submit it. And I have it to where if you accidentally set the wrong thing, you can send another submission. I'm only going to take one, of course, but whatever the final submission is. But if you happen to goof and send the wrong thing, you can simply upload another one. All right, I hope this video helped. Email me if you have questions, and check your email for that information sheet that I'm going to send you for your unknown.